Do you know? On the glorious journey of mankind's pursuit of science, there have been some unknown dark footprints. Those horror scenes that even science fiction movies dare not act in have really happened in human history. Today, we are going to take you into those crazy experiments in the history of science and uncover those chilling truths that challenge the bottom line of human ethics. The second place was to have a woman cross with a chimpanzee, and the first place was to inject human stem cells into a pig embryo to successfully create a human-pig chimera. New viewers can like and subscribe to the little bell so as not to miss the great video. Without further ado, let's get straight to the point. 10th, the Little Albert Experiment. In the long history of the development of psychology, the Little Albert Experiment, jointly conducted by American psychologist John Broadus Watson and his assistant Rosalie Rayner at Johns Hopkins University in 1920, has always been a controversial existence. The subject of the experiment was a 9-month-old baby named Little Albert. Before the experiment began, the researchers tested Little Albert on his emotions and found that he showed no fear of objects such as mice, rabbits, dogs, monkeys, masks, cotton wool, and burning newspapers. This suggested that Little Albert had no natural fear of these objects. When Little Albert was 11 months old, the experiment began. Initially, the researchers placed a white mouse in front of Little Albert and allowed him to touch and play. Little Albert was full of curiosity and joy about the mouse, and there was no trace of fear. Then, when Little Albert reached out again to touch the white mouse, the researchers slammed the suspended iron rod with a hammer behind him, making a deafening sound. This sudden loud noise frightened Little Albert, and he immediately began to cry. This process was repeated many times, and every time Little Albert touched the mouse, it was accompanied by a cacophony of noise. After many experiments, even without noise interference, Little Albert immediately showed a pronounced fear response as soon as he saw a white mouse. This fully showed that Little Albert had closely associated the white mouse with the loud noise and had formed a conditioned fear of the white mouse. More shockingly, however, Little Albert's fear response was not limited to the white rats. On the 17th day of the experiment, the researchers placed a rabbit in front of Little Albert, who also expressed uneasiness and fear. Little Albert showed a similar fear response to a fluffy dog, a sealskin coat, and even a Santa Claus mask with a white cotton beard worn by researchers. This meant that Little Albert's fear had generalized to all furry objects. This experiment caused an uproar in the psychological community, and ethical controversies abounded. Firstly, the experimenter conducted an experiment on Little Albert without telling his mother the real situation, which could cause long-term psychological trauma. After the experiment, the researchers did not try to eliminate Albert's conditioned fear response, which he continued to suffer from for the rest of his life. In addition, video footage suggested that Albert might be showing signs of mental retardation, which undoubtedly further exacerbated the ethical controversy of the experiment and raised questions about its scientific validity. Years later, Albert died of hydrocephalus at the age of 6. 9th, Appleton drug experiments. In the 60s of the 20th century, the United States conducted a series of high-profile experiments, the most well-known of which was the drug test that took place in Holmesburg Prison in Philadelphia, Pennsylvania. The experiment, led by Albert Kligman, a dermatologist at the University of Pennsylvania, aimed to test the effects of various chemicals and drugs on the human body. Dr. Kligman conducted drug tests on thousands of prisoners, applying or injecting a variety of chemicals, including pesticides, herbicides, dioxins, and more to their skin to see the effects of these substances on their skin and overall health. In addition, he used antibiotics, psychotropic drugs, and exposed prisoners to different biological agents to study their effects on the human body. Most of the prisoners involved in these experiments came from low-status groups, many of whom were African Americans. They were tempted by the meager pay, often only a few dollars per experiment, however. These experiments often had serious consequences for their health, including skin burns, scarring, and even psychological trauma. Even more frighteningly, many prisoners were not adequately informed of the nature of the experiments and the potential risks. When the drug trials in the prison were exposed in the 70s of the 20th century, they immediately provoked a backlash from society. These experiments were seen as serious violations of human rights. In 1974, the U.S. Congress passed the National Research Act, which strengthened the regulation of human experiments and required the establishment of institutional review committees to protect the rights and interests of subjects. However, the impact of this dark history on the victims continues to this day. Many of the prisoners who participated in the experiments continued to suffer from health problems after their release. 8th Kola Ultra Deep Drilling Test. In the history of science, the Kola Ultra Deep Drilling Project, launched by the Soviet Union in 1970, is one of the most daring and crazy attempts to explore the interior of the Earth. In the mid-20th century, with the continuous development of geological science, scientists became interested in the internal structure of the Earth. At that time, the United States launched the Mohole Project, which tried to drill through the Earth's crust and reach the mantle to study the Moholovich discontinuity between the crust and the mantle. Not to be outdone, the Soviet Union, a rival of the Cold War, decided to launch its own ultra-deep drilling program on the Kola Peninsula. On May 24, 1970, the Kola ultra-deep hole was drilled to a depth of 15,000 kilometers. Drilling was carried out by the Soviet Scientific Research and Development Center Nedra, located about 10 kilometers west of the city of Zapolyarny in the Murmansk region. The diameter of the upper part of the borehole was 92 centimeters, while the diameter of the lower part was reduced to 21 centimeters. When drilling to a depth of 7,000 meters, the drilled bit entered the softer layered rock, causing the borehole wall to collapse and the drill string to get stuck or fractured several times. After each accident, the engineers had to reseal the section, adjust the drilling tools, and then continue drilling. Despite all the difficulties, in June 1979, the Kola hole broke the U.S. record of 940 meters in the Bertha Rogers ultra-deep well in Oklahoma, becoming the deepest hole in the world. In 1983, the hole was drilled to a depth of 186 meters in preparation for the 1984 Interregional Geological Congress in Moscow. Drilling was temporarily suspended on 27 September 1984. Drilling resumed. But soon after, another drill string fracture occurred. Engineers restarted drilling from a depth of 7,000 meters, eventually reaching a maximum depth of 12,262 meters in 1990. However, as the depth increased, the ground temperature also rose sharply, far exceeding the expected 100 degrees, reaching an astonishing 180 degrees, which posed an unprecedented challenge to the drilling equipment. The Kola ultra-deep drilling program has yielded fruitful scientific results. In the strata at a depth of more than 9,500 meters, hydrogen-rich rocks were found, and the mud gushing out of the borehole even showed a boiling state due to the high hydrogen content. In addition, the project revealed the existence of a large number of microbial life in the Earth's crust, revolutionizing people's perception of the depth of life. However, due to financial difficulties and lack of state support, the Kola ultra-deep drilling project was halted in 1995. In 2008, the borehole was officially abandoned. The equipment was dismantled, and the above-ground buildings were damaged. Today, all that remains of the once glorious drilling site is a wellhead sealed with a metal cap, only 23 centimeters in diameter, as if it were a scar left by human exploration into the depths of the Earth. 7th Two-Headed Dog Experiment. In the 50s of the 20th century, the Soviet scientist Vladimir Dimitrov conducted a series of experiments that shocked the world. He succeeded in transplanting the head of a puppy into the body of another adult dog, creating the two-headed dog. Vladimir Dimitrov is known as a pioneer in the field of organ transplantation. back in 1946. He successfully performed the world's first heart-lung transplant on a dog in 1952. He successfully performed the first live dog heart transplant operation, and the dog survived for 5 months. These achievements made him famous in the field of organ transplantation. However, he didn't stop there and set his sights on a much bolder experiment, a head transplant. In 1954, Dimitrov began his two-headed dog experiment in a laboratory in Moscow. He carefully selected an adult German Shepherd as a host and transplanted the head and forelimbs of a puppy into its neck. During the procedure, he connected the puppy's blood vessels to the adult dog's circulatory system, ensuring that the puppy's head received adequate oxygen and nutrients. However, the puppy's esophagus was not connected to the adult dog's stomach. The puppy's head could lick milk, but it was not really able to take in nutrients. In the short time following the surgery, this two-headed dog showed amazing vitality. The puppy's head was able to respond quickly to external stimuli. However, due to immune rejection and surgical complications, these two-headed dogs often only survived for a few days. According to relevant reports, Dimitrov had conducted several similar experiments. The longest survival time reached 29 days. Dimitrov's experiments caused an uproar in the scientific community, and the voices of ethical controversy were heard endlessly. His use of animals as experimental subjects and such extreme surgeries resulted in great suffering and short lives, which was considered extremely inhumane by many. 6th Zombie Dog Experiment. In the history of science, in 2005, a research team from the University of Pittsburgh in the United States conducted a high-profile experiment known as the Zombie Dog Experiment. Researchers at the University of Pittsburgh selected healthy dogs as test subjects. They first drained all the blood from the dog's body through drainage, causing the dog to enter a bloodless state. The researchers then injected an ice-cold saline containing oxygen and sugar into the dog's vasculature. This cryogenic saline worked by lowering the dog's body temperature and slowing down the rate of its metabolism, thereby reducing the need for oxygen in the tissues. In the process, the dog's body temperature was lowered to near freezing, its heartbeat and breathing stopped completely, and the brainwave activity disappeared without a trace, entering the state of so-called clinical death. This state lasted for about 3 hours. 3 hours later, the researchers began the process of resuscitation. They expelled all the saline from the dog's body and returned to fresh blood. At the same time, electric shocks were used to stimulate the heart in an attempt to restart the heartbeat. After a series of intense resuscitation measures, some of the dogs successfully regained their spontaneous heartbeat and breathing, showing signs of life again. The results of the experiment showed that the dogs that were successfully resuscitated were basically the same in terms of behavior and physiological function as before the experiment. However, there were also some dogs that had varying degrees of organ damage and neurological dysfunction. The researchers speculated that this might be due to factors such as hypothermia, length of time in a bloodless state, and the techniques used during resuscitation. 5th Tuskegee Syphilis Experiment. In the United States in the 20th century, a medical scandal broke out and shocked the world. The Tuskegee Syphilis Experiment. At the beginning of the 20th century, the rate of syphilis infection among African American communities in the southern United States remained high. To study how syphilis could develop without treatment, Tuskegee University launched the study in Nitern-Tundy in Macon County, Alabama. A total of 309 African American males infected with syphilis and 2 Hun-Main males without syphilis were subject, all of whom were farmers living on a budget. The researchers promised to provide them with free medical checkups, food, and funeral insurance. At the beginning of the experiment, the standard treatment for syphilis was the use of arsenic and mercury preparations, but the side effects of these drugs were significant and their efficacy was limited. However, by the 40s of the 20th century, the discovery of penicillin led to a major breakthrough in the treatment of syphilis. Despite this, the experiment's researchers chose not to give the subjects penicillin to the treatment. and even prevented them from receiving treatment elsewhere. They hoped to gain insight into the long-term effects of syphilis on the human body if it was not treated. During the experiment, the researchers regularly examined the subjects under the guise of special treatments, and the items examined included invasive procedures such as blood draws and lumbar punctures to obtain research data. The subject had been kept in the dark and mistakenly believed that he was being treated. The Tuskegee syphilis experiment was devastating for the subjects and their families. By the time the experiment was revealed in 1972, 28 people had died of syphilis, and about 100 had died of complications of syphilis. 40 people had their wives infected, and 19 children had contracted syphilis at birth. The incident caused a strong outrage in society, and it became one of the most serious ethical scandals in the history of medicine. Subsequently, the victims and their families filed a class action lawsuit, and the U.S. government eventually agreed to compensation and provided lifelong medical care to the survivors. In 1997, U.S. President Bill Clinton officially apologized to the victims and their families on behalf of the government. Fourth experiment on the effects of radiation on pregnant women. In the 50s of the 20th century, the United States made rapid progress in the fields of nuclear technology and radiation medicine. With the successful development of nuclear weapons and the development and utilization of nuclear energy, the U.S. government and the scientific community had begun to pay special attention to the effects of radiation on human health. In particular, they wanted to know what the effects of radiation would have on pregnant women and their foetuses in order to assess the risks to this sensitive group in the event of a nuclear accident or medical radiation. In 1950, the U.S. Atomic Energy Commission conducted a number of studies at the University of Iowa to investigate the effects of radioactive iodine on pregnant women and newborns. One study was to study how radioactive iodine crossed the placental barrier by taking 100 to 200 microcuries of iodine-31 orally to a pregnant woman, followed by dissection of the aborted fetus. In another study, 25 newborns were given iodine-130 orally or injected. The researchers then collected samples of thyroid tissue from the babies to assess how radioactive iodine was absorbed and metabolized in the newborns. It is worth mentioning that the subjects were not fully informed of the true purpose of the experiment, the specific process, and the possible health risks. Pregnant women and newborns are among the most vulnerable groups and deserve special protection. However, these experiments put them at risk of radiation exposure, violating the principle of do no harm in medical ethics. After the experiment, the subjects were not able to receive the necessary treatment, and the effects on them continued for the rest of their lives. Many babies were born with malformations. Their thyroid function was affected, and the risk of cancer was greatly increased. Third, human experiments in Nazi Germany. During World War II, Nazi Germany carried out a large number of brutal human experiments on prisoners in concentration camps. These experiments were conducted primarily on Jews, Gypsies, Soviet prisoners of war, and the disabled, with extreme cruelty and complete disregard for humanity. The Nazi regime promoted racial superiority, treating Jews, Gypsies, etc. as inferior races and systematically persecuting them. Here are some typical experiments. Cryogenic experiment. In Dachau, Nazi doctors exposed prisoners to extreme cold or immersed them in ice, water to study how the human body reacted to hypothermia and how to rewarm it. Many subjects died during the experiment or suffered permanent damage. High-altitude experiment. In order to simulate the conditions of high-altitude flight, prisoners were placed in low-pressure chambers to simulate altitudes of up to 200 meters, resulting in many deaths due to lack of oxygen or decompression. Chemical weapons experiment. In the Sachsenhausen concentration camp, prisoners were exposed to chemical agents such as mustard gas to study their effects on the human body and how to treat them. Subjects often suffered severe chemical burns and even death. Sterilization experiment. The Nazi government pursued a policy of forced sterilization, sterilizing hundreds of thousands of people. In Auschwitz and Ravensbrück, doctors sterilized prisoners using X-rays, surgeries, and drugs, resulting in many serious injuries. Seawater experiment. At Dachau, prisoners were forced to drink only treated seawater as a way to study the drinkability of seawater. Subject was on the verge of death due to severe dehydration. These horrific experiments resulted in the death or permanent damage of a large number of victims. The survivors were also traumatized physically and mentally, regarded as one of the darkest pages in the history of medicine. The Nazi human experiments prompted the post-war international community to develop the Nuremberg Code, which regulated ethical issues in medical experimentation. Second human-ape hybrid experiment. In the 20s of the 20th century, Soviet reproductive biologist Ilya Ivanovich Ivanov conducted a series of highly controversial experiments in which he tried to achieve the crossbreeding of humans with chimpanzees through artificial insemination technology, so as to create the so-called human-ape hybrid offspring. Ivanov was a pioneer in the field of artificial insemination, and he had successfully applied this technology to horse breeding, which had greatly improved the efficiency of breeding. On this basis, he came up with an even bolder idea to explore whether there was a possibility of reproduction between humans and other primates. In 1926, Ivanov traveled to Africa and tried to inject human sperm into the body of a female chimpanzee through artificial insemination. However, these attempts ended in failure, and the conception was unsuccessful. In 1929, Ivanov changed tactics and planned to inject sperm from chimpanzees into human women. He recruited 5 female volunteers in the Soviet Union who were willing to participate in the experiment. However, the experiment was terminated due to the death of the male chimpanzee needed for the experiment at a critical moment. Ivanov's experiments caused an uproar in the scientific community and even the entire society, causing huge ethical controversy. Many believe that this attempt at cross-species hybridization is a serious violation of human moral and ethical norms and a great desecration of human nature. In 1930, as the situation changed, Ivanov was arrested and exiled to Almaty, Kazakhstan. He died in exile in 1932 at the age of 61. First human-animal chimera experiment. In 2016, scientists made a major breakthrough in the field of stem cell research by injecting human stem cells into pig embryos to create pig embryos containing human cells, also known as human-pig chimeras. Scientists hope to use this to grow human organs in pigs that could be transplanted. The research team at the Salk Institute for Biological Studies in the United States first used CRISPR gene editing technology to delete the genes responsible for the formation of specific organs in pig embryos, thereby creating genetic vacancies. They then injected human-induced pluripotent stem cells into these edited pig embryos. These iPS cells had the potential to differentiate into various cell types. The researchers implanted human stem cells into more than 500 pig blastocysts and implanted these chimeric embryos into sows. The embryos developed in the sow's body for 3 to 4 weeks before being removed for analysis. The results showed that the human cells successfully survived in the pig embryo and began to differentiate, albeit in a relatively small proportion. Although human cells successfully survived and began to differentiate in pig embryos, their proportion was still low. The researchers believed that this result showed that human cells could be grown in pig embryos, but there is still a long way to go to achieve the cultivation of complete human organs. They plan to increase the proportion of human cells in the chimera by optimizing cell types and timing of injections. As soon as the study of human-pig chimera was published, it sparked a wide range of ethical discussions. First, If human cells make up a large proportion of an animal's brain, it could lead to animals possessing human cognitive abilities or consciousness, which would blur the boundaries between humans and other species. At the same time, chimeras may also pose unknown biosafety risks, such as the spread of novel pathogens. In response to these concerns, the National Institutes of Health suspended funding for human-animal chimera research in 2015. Recently, however, the study was restarted. I wonder if there will be pigs who can speak human words in the future?